Today we're going to be discussing the cell cycle. So what happens to cells during division? What is actually happening at those checkpoints? What is controlling and regulating them? And also, why and how do cells divide? How do cells know when to divide? That was a question scientists asked before they knew what was regulating them. So there had to be some sort of regulatory mechanism because cells are not always dividing. But how do cells know when to divide? So if you look at this image here, these are cells in a petri dish that were grown. This tissue was scraped out here in the middle, creating free space, and the cells along the edge continued to divide until this artificial wound was healed. And that's what cells do. But how do cells know when to stop and what's controlling that? That's the question scientists had. So when an injury occurs, what I want you to write down is when an injury occurs, the neighboring cells, so the cells that are bordering that injury, kind of like here in this example, they're going to be stimulated to rapidly divide because they want to heal. They want to protect the body. Remember, homeostasis. So they're going to divide. Then, when the healing process is almost complete, division slows down. So that rate of division slows and controls on growth are restored. So the body is somehow signaling for those cells to stop. And normal activities in the cell return. By normal activities, what phase of the cell cycle is that going to be? So the cells have a way of turning division on full blast and slowing it down and turning it off. So the cell cycle is controlled by regulatory proteins, and these are found both inside and outside the cell. It's all found within our body, but inside the cell and outside the cell, meaning hormones and proteins. Internal regulators are proteins, which we're going to learn about, called cyclins, that act as checkpoints, only allowing the cell to proceed through the cell cycle if certain events are met. External regulators, they direct cells to speed up or slow down in the cell cycle. So example are growth factors, which stimulate division in embryonic development. So growth factors occur naturally in the developing embryo, so in the baby that is forming, because we want that division to occur rapidly, and it also occurs naturally in adults. Growth factors can also be given artificially or as an injection to adults who don't produce enough of it. So we'll talk about growth factors. Again, here, these are the cells that will be signaled to divide and fill this open space until there's no more room for division. So during the cell cycle, there are three major checkpoints. Remember, we talked about how they're kind of like a lag. They occur to ensure that this process is happening correctly. The first major checkpoint is the cell growth checkpoint. Has the cell grown enough? Is it large enough to divide? Is there enough cytoplasm, cell membrane? Are the organelles divided? So this checkpoint is the first checkpoint in the cell cycle, G1. It occurs towards the end of G1, and it's going to ensure that the cell is large enough. It has proper proteins for S phase, so it needs helicase and DNA polymerase and ligase. All of those proteins must be synthesized as well as all the organelles before it can go. If it doesn't meet this criteria, the cell goes into G0 until it's ready to divide. The second checkpoint is the S checkpoint, or DNA synthesis checkpoint, which will, and I would draw this if you haven't in your notes already. Pause the video and draw. So the second checkpoint is S during S phase or shortly after it has occurred, it's going to check whether the DNA has been replicated correctly, so ensuring that DNA polymerase has checked and correct any mistakes, so there's no mutations. And if so, the cell continues on to mitosis. If it doesn't meet this, that is when the cell goes into apoptosis, or programmed cell death. Mitosis checkpoint. So this is a third checkpoint, and it occurs during mitosis. It checks whether mitosis is complete. So has the nucleus and all the DNA been divided? And if so, the cell continues to divide in cytokinesis, and the cycle repeats. So it's going to check before cytokinesis. And again, if the cell doesn't meet the criteria, it will go into programmed cell death, or apoptosis. So cyclin was the first regulatory protein that was discovered by scientists to regulate the cell cycle. And it's a cycle, so they named it cyclin. That is the protein, how it got its name. And so these cyclins regulate the timing of the cell cycle in eukaryotic cells. And we're only learning about regulation in eukaryotes. So they will control how quickly the cell goes through the cell cycle. And cyclins, you'll learn more about if you take AP Bio, but they form a complex with cyclin, or a CDK cyclin kinase complex. So it's two proteins, and kind of looks like the ribosome. I didn't mean to make it look like a ribosome. I'll change the shape a little bit, but it's a lock and key like all enzymes are. And this complex, when it's bound together, will allow I'll draw a different color, but it will allow the cell to continue through the cell cycle, and without it cannot. So you'll learn more about that in AP Bio. We'll learn a little bit more about it in our discussion when they appear. So in our discussion on Axis, you guys are going to be evaluating the data in these graphs of when cyclin is produced. So when is it produced? And I want you to analyze these, but you're going to discuss on Axis, and we'll talk about it more after you guys fill in your discussion. And these are just some questions to think about, because again, you're going to be coming to these conclusions in your discussion and talking about it with your peers on Axis, so we'll talk about it more. But I want you to start thinking about these, looking at the graph. What is the data telling you about the cyclin protein, and how does it regulate the cell cycle? So this is something I want you to do on your own. Really look at what is the independent variable, what is the dependent variable, what type of cells are being looked at. So be sure to really look at the data and see what it's telling you. So cancer, how does this relate to the cell cycle? Cancer occurs when cells do not respond to the normal regulatory signals of cells. So most cells will respond to these regulators, both internal and external regulators, such as cyclin or growth hormone, but there are some cells, due to mutations, that do not. And the cell cycle is then disrupted. Cancer cells, if you look here, and I would copy this chart, but they continue to grow and divide. So that's fact number one. They will continue to grow and divide, and that mass becomes a tumor. And tumors can be malignant, meaning harmful or deadly, or benign, meaning not harmful or deadly. And most benign tumors are just removed and they're fine. The malignant tumors are the ones where treatments like radiation and surgeries or chemotherapy is used to treat these cells if it's malignant. And then variations in size and shape of the cells. So if you notice, compared to normal cells, cancer cells have very rough shapes. They're not smooth. The cell membrane and the cytoplasm are not as uniform as a normal cell. And cancer cells also have a larger nucleus than normal cells, and it's much darker. They also can have abnormal number of chromosomes, either too many or too little, and they will not stop dividing even if they have reached a boundary with another cell. So most cells have contact inhibition, meaning they will stop growing when they come in contact with another cell. Cancer cells do not. And if you look here, one of the reasons why cancer is so deadly is because cells are constantly dividing. You can see the cells in anaphase. We know these are cancer cells because their nucleus is larger and they don't have as much of a uniform shape. And cancer can be deadly because it can spread. So these cells that were in the tissue have now gone into a blood vessel, and now this cancer cell can spread to another area of the body. So that's why cancer can be harmful if not treated right away or not caught on time. So cancer cells. Normal cells respond to growth factor. So if we put normal cells in a petri dish, one with growth factor, one without growth factor, what would we expect to see? So normal cells with growth factor. Growth factor, if added, is an external regulator that allows cells to divide rapidly. So we would see normal cells dividing. But once they came in contact with each other, they would stop. And remember, cells only come from existing cells, so there would have to have been cells in this petri dish already. And it would go until there was no more space for division. Remember, they're dividing through mitosis. Without growth factor, normal cells respond by not growing, or at a very slow rate. So we wouldn't see, if we only put in a small sample of tissue, that's all we would see. Even after a few days, there would not be any division. However, cancer cells with or without growth factor will divide. And again, cancer cells have weird shapes. They have a larger nucleus. And they will even grow on top of each other, creating that tumor. They do not have any contact inhibition. So that can be seen. Cancer cells will grow with or without growth factor. Unlike healthy cells, which will not grow if there's no growth factor, cancer cells will, no matter what. And I would copy this into your notes. So just to review, they are dividing through mitosis, which is producing genetically identical cells. They are diploid, meaning they have two sets of chromosomes, one from mom, one from dad. And this can take place throughout an organism's lifetime. But mitosis, as well as the cell cycle, is regulated.